All right, finally, we are going to go through the easiest part, I promise, of the kidney filtration system, which happens in the glomerulus. So we call this glomerular filtration. And in this section, we're going to go through the structure of the glomerulus and talk about the pressure gradients that exist there that are going to drive filtration. So I want you to think about the glomerulus actually as a filter in the same way there's filters in fish tanks, or more simply, a strainer that you would use to drain something like pasta. The glomerulus is just a collection of capillaries with fenestrations in it, so just little holes. And the blood is going to come in. Remember, it's going to go from the aorta to the renal arteries and then get squeezed into smaller and smaller spaces until it gets to the smallest levels called the arterioles. So there is an afferent, which means going towards, and an efferent, which means going away from arteriole. And this is going to be a very high pressure system. So imagine things entering here at a really high pressure, and then there's holes in this capillary bed. And then the outlet is going to be a little bit smaller than the inlet. So that means more blood is going to come in a larger area, and then it's going to have to squeeze through a smaller area to leave. This is going to set up the absolute best conditions for filtration, meaning that small substances can leave the bloodstream and enter the next thing that we talked about, the renal corpuscle. The renal corpuscle is going to encompass both the glomerulus and Bowman's capsule. Bowman's capsule is the first area that is going to catch these filtrate products. So the first products are really just going to be anything that's small and can make it through these fenestrations. So it doesn't necessarily mean that it's going to be a waste product right away. What it simply means is that it's going to be small enough to fit through the holes. Very simply, like when you drain your pasta, the pasta is retained in the strainer because it cannot fit through those holes, whereas the water and maybe any salt that you added to the water or oil, those things will leave just naturally because they can fit. So those we're going to refer to as the filtrate. And the filtrate is made up of waste products, but also some just very small things that we might not want to get rid of, meaning that we can trade things back and forth through this complicated network of blood vessels that wraps all the way around these different parts of the renal tubule. So blood enters the glomerulus. Small things that fit through the holes will enter now Bowman's capsule and then move throughout the tubule. So we'll see that we are going to get things that are just tiny, mainly water and ions, but also some waste products like urea. So things like nitrogenous waste that we do want to get rid of. And this is the process that we're going to be talking about in this lecture. So just the movement of things out of the glomerulus and into Bowman's capsule, and then it will traverse the renal tubule, and really anything that's going to make it to the collecting duct will be incorporated into the urine depending upon which hormones are present. So we'll talk about those in the next lecture for secretion and ultrafiltration. So right now we're just talking about what's going on at the level of the glomerulus and how this is the perfect system to drive a force for filtration. So as we mentioned, there are these two separate arterioles, and if you had watched the initial anatomy and physiology video, I said that these arterioles are going to be really the place where the physiology happens. So we have the afferent arteriole, afferent coming towards, and then we have the efferent leading away from arteriole. And arteriole literally means really small artery. So the one that leads to the glomerulus and the one that leads away from it. So all of the products in the bloodstream, again, they're going to be squeezed into smaller and smaller vessels until they get to these areas of physiologic action, these arterial areas. So the blood is going to come in at a really high pressure in this afferent arteriole, enter the glomerulus, here's our glomerulus, which is filled with little fenestrated holes. That's it. So this is the first process, glomerular filtration, and it's really just a simple process where anything that's small enough to make it through this hole will then enter Bowman's capsule and then traverse through the renal tubule where it's further acted upon. The other processes that we'll talk about that are way more complicated, we have the process of reabsorption. So remember when we were learning about digestion and all the food products get down to the small intestine's job is to absorb products, correct? It's absorbing things like water and nutrients, and it's absorbing them right into the bloodstream. So if we think about this, all of this blood entering the glomerulus, that contains absorbed nutrients and absorbed products. Absorption means it's absorbed into the bloodstream. Reabsorption describes the process of something having reached the renal tubule. So here we're on process 2, reabsorption. And remember, everything flows high to low concentration. So it might be that a lot of water entered the renal tubule, but we don't want to lose all of it. So it might be reabsorbed if the amount of the filtrate is lower in the capillaries that wrap around the renal tubule. We call these the peritubular capillaries. Maybe it's an ion, maybe it's potassium. Stop, dogs are allowed to go for walks. It's okay, sorry about that. We don't like other dogs going by the house, and we definitely don't like the mailman. All right, so these products were already absorbed into the bloodstream. Now they've made their way into Bowman's capsule and the tubule. And if there is this concentration gradient where the concentration of things is higher in the nephron than it is in the blood, those products can move back into the bloodstream. So that's called reabsorption because it was already absorbed through normal digestive processes, but it was lost to Bowman's capsule. When those things move back into the blood, that's called reabsorption. So it's brought back into the bloodstream. The third product is secretion, and we'll talk about that in the ultrafiltration chapter. If there's anything extra in the blood, if things are really high in the bloodstream and we want to get rid of them, remember high to low concentration. So let's say this person has acidosis. It means there's a high amount of hydrogen ion in their bloodstream. We can have those products added to a specific site in the renal tubule to maintain electrolyte balance, to maintain acid-base balance. So that's called secretion, and that they're added later on into that process. And the fourth and final thing is the easiest one. That's just excretion. So once all of these fine-tuning adjustments have been made to this initial filtrate, which just made it through those small little holes in the glomerulus, the leftover product in a healthy system is referred to as a concentrated urine, which will then be excreted, meaning that it's going to move to the bladder, which can then be emptied in the micturition response. So excretion is going to be the products of filtration, of glomerular filtration, minus anything that's reabsorbed back into the bloodstream that wasn't meant to be gotten rid of. It was brought back into the blood, plus anything extra that was secreted by the body into the urine to get rid of in a different way. So we're going to have these initial products of filtration, then we have the more complicated reabsorptive processes, plus the secretion. So, sounds fun, right? It is. I actually like the kidneys. They're it's complicated. It's really interesting how much goes into really making a concentrated urine. So again, we're just going to concentrate on this for the remainder of these slides. The way that we measure how fast, or really it's a pressure, so how much filtration is going on, is called the glomerular filtration rate, or GFR for short. So you'll see GFR on a lot of these slides. The glomerular filtration rate is the rate at which products are being squeezed from the glomerulus into the Bowman's capsule and renal tubule. The way that we can measure this is with a clearance test. And clearance tests are pretty non-invasive unless you happen to go into somebody's office when they're doing it. A clearance test involves somebody collecting their urine, usually for a 24-hour period, sometimes for 36 hours. And they're just going to collect everything that they excrete in a plastic jug. That will give you a way to look at throughout a day or a day and a half, a couple of days, how much of different products is excreted in the urine. So we want to confirm that there is a concentrated urine containing the waste products that are necessary for a nice working body without toxemia or any other issues. And one thing that I've mentioned early on in the anatomy lecture is this process called creatinine. So you've probably heard of creatine. So creatine is something that you can buy at health food stores or probably even Wegmans, and it's added to different drinks. And what it essentially does is allow a little bit more energy to be taken up by the skeletal muscle. And you might know that skeletal muscle hypertrophy can be increased when you increase things like maximum weight and maximum reps. So there's usually a point of muscle fatigue where somebody has their max weight. If you could squeeze in one extra rep, like one rep literally of max weight, then you're going to get greater muscle hypertrophy, which means that the oxidative capacity of muscle will go up over time. So creatinine is something that is utilized to allow this extra energy to exist by adding this extra energy source to skeletal muscle to allow muscles to work a little bit further past their typical exhaustion capacity. A breakdown product of creatine is creatinine. And one thing you should know about creatinine is that it has no physiologic activity in the body. So that means any creatine, so any skeletal muscle creatine that's broken down into creatinine, any creatinine that's created is not going to go anywhere in the body because it has no function. That means it's going to be peed out. Any creatinine should be urinated out. So that gives us a really good thing that looks like read, but it says peed. So this will be excreted, and this should be a really standard measurement. Everyone with nice working kidneys should excrete a standard level of creatinine. So any creatinine that enters the glomerulus will be filtered by the glomerulus and then be excreted by the kidneys. So you can look at how much urine creatinine exists to determine really easily if the kidneys are working, and then also that gives you a readout of what the rate is of filtration. So the amount of creatinine excreted in urine in a minute should be the amount of creatinine filtered at the glomerulus. So we've got our plasma creatinine, urine creatinine. So this is something that can be figured out mathematically, and it's a really easy measurement. Just if you're interested in some fun facts, I like to give fun facts every once in a while. One of the ways, I'm going to reset my drawings, that I used creatinine in the past is in a study that I did in graduate school. We were looking at heavy marijuana users, so MJ positive. And in order to be eligible for the study, heavy marijuana users, they had to smoke between 3 to 6 blunts a day. That's a fairly large amount of marijuana. So we ran these studies during the day. Smoking that much marijuana generally meant that those individuals were free throughout the day to visit the lab. And one thing that heavy marijuana users report is that it's hard for them to quit. So it's always been supposed that there was a withdrawal syndrome associated with marijuana, but it hadn't really been scientifically verified. People just seem to be cranky and irritable, and they report that there's trouble sleeping. And the easiest way to make that go away, if there's withdrawal syndrome, is to re-administer the drug. So what we did was study this. We had heavy marijuana users come in, and they had to abstain from marijuana. It's what we found out is that about 2 to 3 weeks after stopping is when the symptoms of withdrawal are most intense. And then once people get over that 3-week kind of hump, the symptoms dissipate and it becomes more easy to deal with withdrawal effects, which then become very, very minimal once you're out past 4 weeks. So that's what we found out, was that it was something that was scientifically verifiable. There are a large amount of sleep disturbances. We had them in the sleep lab. So we did a lot of science on them, which was cool. But how do you make sure that these heavy marijuana users are abstaining from marijuana? Well, the simple test is a urine test. And if you're familiar with drug tests, you might know that you can test positive for marijuana in a urine test for days afterwards. If it was somebody's first experience with marijuana, they are marijuana naive, they usually have a positive test for about 3 days afterwards. And that's just one time. If it's a regular marijuana user, because marijuana THC is stored in the adipose tissue, it's released really slowly throughout the day, especially when you break down fat. So when people do things like go up the stairs, it's released really slowly anytime there's lipolysis. So what you see is that regular marijuana users usually have a positive test for about 3 weeks. That's normal in a normal weight individual. There is a world record in somebody with a higher amount of adiposity of a 3-month window between the last time they smoked marijuana and a positive test. So it's really dependent on the amount of adipose tissue. And that is something that varies in individuals. So if we have these individuals into the lab for these studies, we need to make sure that they're not smoking, that they're being compliant with the study. Otherwise, their data wouldn't be valid and we wouldn't be able to verify anything that we found. We know that they're going to have a positive urine test, right? Because we have them for a couple of weeks afterwards, even if they're abstaining. So what we looked at when we sent off the urine was a THC to creatinine ratio. So what you should see is over time, if people are truly abstaining, their THC levels go down where their creatinine levels should remain stable. So does that make sense? I hope it makes sense. So not only is it a nice measure for getting the actual rate of filtration, it's also a really stable baseline so that you can actually measure the concentration of exogenous substances in the urine as well, because we know creatinine should be standard. So that's my fun fact about THC versus creatinine, and I hope you learned something about drug tests. Just another fun little fact, there is no way to beat this by buying anything special in a store. There's only one way to have a negative drug test when you're positive for a substance, and I'm not going to tell you what it is. I'm sorry. Okay, so back to our glomeruli. This is a photograph, an electron micrograph image of glomerular capillaries, and some things that I want to point out to you are A, these fenestrations. So fenestrations, they're just little holes. So this picture in B, these areas of holes, those are our fenestrations. So they work just like a sieve, which is a fancier name for a strainer, usually with smaller holes. So anything that can fit through these fenestrations will just go through because of the pressure gradients. So that means things like small ions can fit through, like sodium, potassium, but larger things shouldn't be able to move through these spaces. The other thing that aids in filtration are podocytes. Podo means foot, and cyte comes from cyto, which means cell. So that's what you're looking at in A. So this is a close-up picture in B of just these fenestrations, and in this A column, this is a podocyte cell. And these podocytes, they wrap their foot processes around and around and around. This is one podocyte right here, kind of like an octopus wrapping its tentacles around something. And it wraps it around these capillaries so that in between these podocytes, which then you can see how they're almost like fuzzy little dendrites, they create things called slit pores. You see them here? So podocyte and then these little extensions. So those are going to overlay these fenestrated endothelial walls of the capillaries so that we have two things that substances have to make it through. This is a beautiful podocyte right here. It first has to make it through these fenestrations, and then it has to squeeze through the slit pore made by the podocytes. So just to give you an idea, if we're going back to the strainer example, if I draw my strainer, that looks really good right here. It's in my sink. It looks like the little teacup for Beauty and the Beast, but it's not. But I'm going to draw it like him anyway. So there's holes in this strainer. So if I just made some Kraft macaroni and cheese, and I put my little noodles in here. I like the shells more than the elbows, just FYI. They're way better. They hold the cheese as more of a vessel. It's a personal preference. So the noodles will be retained in my sieve because they are too big to make it through these holes. The water, though, is going to make it through because water is small. So water is going to make it through these strainer holes really easily. Using the example of ions, if I were to have my strainer still in my kitchen sink and I took my salt shaker out and shook some salt into this strainer, those little pieces or crystals of salt, those would go through the holes too because they're tiny. So that's your example of ions, right? So we know little things like water and ions, they can move through because they're small. In this strainer, I'm going to draw another picture because I've made that kind of busy. So here's my strainer again. We've got our little holes in it so we know water and ions can go through. If we look at the actual size of albumin in relation to these little fenestrations, using our sieve example, that would be like putting a hard-boiled egg into the strainer. That egg is way too big to make it through the hole, and that's our protein. So protein is too large to make it through those holes. If you see protein leaving through the urine, that's called proteinuria, and that's a sign of glomerular damage. It means something has made these holes really big and now protein's getting through. What's even larger is things like red blood cells. That would be like putting a hamburger patty into my strainer. Hamburger. So that's really big. And if you see blood in the urine, that's called hematuria, blood in the urine, that means that these holes are so big that red blood cells make it through. So that'd be like having a hole in your strainer so big that a hamburger patty can make it through. So this is just a size thing. So just keep in mind things that are small, they can move really easily through these fenestrations because of the size. Simple, right? The other thing that is going to allow this movement for filtration is these pressure gradient differences. These are all parts that we're going to talk about in a little bit. So P is like podocyte. We'll talk about all those in a little bit. Don't worry about it right now, but do worry about this. AAEA, so that's our afferent arteriole, efferent arteriole. These are the macula densa cells there. And PT stands for proximal tubule, the closest part of the tubule to the glomerulus. So what I want you to look at right now is just the size. We've got the afferent arteriole right here, and then we have the efferent arteriole. So blood's going to come in this afferent arteriole at a really high pressure. It's going to be squeezed through these tiny little blood cells, and eventually, or blood vessels, those filtrate products will make it into the first part, this Bowman's capsules part of the proximal tubule. Then the blood that is now empty of its filtrate, empty of little products that have made it through those fenestrations, will then leave through the efferent arteriole. It's kind of hard to see in this picture, but we'll see it in some other slides too. One thing that helps drive this pressure gradient is that the pressure is really high in that the afferent arteriole is larger in size than the efferent arteriole. So that would be as if we were walking into a room and the inlet was a double door, and the whole class has to get into this room through double doors. We're going to be able to fit people in pretty easily. But then the outlet is just a single door, right? That means we're not going to be able to fit as many people through at the same time, and there's going to be more backup within the room. So there's going to be some limitation to the speed or the rate at which things can move. So this size difference between the afferent and efferent arteriole is going to create a high pressure situation within the glomerulus that will drive the ability for substances to physically leave the bloodstream. If this pressure is really low, if this was a low pressure system, let's say this person's going into shock and there's not a lot of blood flow getting to the kidneys, and actually you see that when the kidneys are really failing, it's called anuria, which means there's no urination. That means the blood pressure is so low that nothing is leaving the bloodstream and then being incorporated into urine. That creates a really toxic situation, toxemia. That means toxic substances in the blood, which would then eventually lead to death, because you cannot have a pH outside of this little window around 7.4. Biochemical reactions cease to occur. So if you retain too much hydrogen ion or bicarbonate, you cannot have biochemical reactions occur. So it's this nice high pressure situation, which will be controlled also by the fact that each of these arterioles has sphincters associated with it. So that means we can regulate the pressure in this glomerulus to ensure that we don't enter a state of anuria or a state of polyuria where there's just watery urine not filled with waste products. So we need to maintain a nice stable rate, this glomerular filtration. So this is very simple. Like I was showing you, it doesn't seem simple when you look at it with big words. That's why I use my sieve example. So when things are really small, they have a perfect ratio. They're 100% filterable, which means that's a 1.0 filterability score. So things like water, sodium, glucose, those are filtered really nicely. Just a little note, you shouldn't have a lot of glucose in your urine. That is a sign of diabetes because your body loves glucose, right? We run on it. So if you have sufficient insulin, then the glucose will be moved into the tissues that need it. So what we call urine that has a high amount of glucose in it, we call that a high specific gravity urine, and that has to do with this molecular weight. So the molecular weight of glucose is relatively high relative to sodium and water, so that would mean that there's very sugary urine. It's filtered really well, but in healthy individuals, there shouldn't be a high amount of glucose. There should really be no glucose in the urine because the body runs on it. Inulin, and then we get to larger like globular proteins like myoglobin, albumin. Those things are not filtered really easily. So like myoglobin, we see filtered in the urine in cases of rhabdomyolysis when there's an abnormal breakdown of skeletal muscle. That's a condition that you have to be hospitalized for. That's really dangerous. And then proteinuria, when there's a high amount of albumin, which is a really large molecular weight, that is a very low filterability quotient, which means things like proteinuria, those are pretty dangerous as well. That indicates that there's damage to the glomerulus. And that's really the rule in pathology. Anytime there's damage to the glomerulus, there's going to be damage to filtration. So that means things are going to move out more easily. Another thing, just because of the way that proteins are charged, it's called the Danon effect. We'll talk about it more in a little bit. But the Danon effect states that proteins are negatively charged so that they can retain the components of the plasma. And that's really important to keep things where they're supposed to be inside of vessels, because we know things like capillaries are really leaky, and there has to be a way to keep everything inside of leaky vessels. Otherwise, it's just going to spread to whoever it wants to. So we want to house things or keep things that are appropriately necessary in the plasma. Just for your own knowledge, Positive substances, they are filtered more readily than negatively charged because, again, things like protein, it's negatively charged. We try to maintain those negative charges so that we have stable plasma levels. Whereas negatively charged substances, they have a lower filterability ratio. So cations are positively charged because everybody loves cats. Cats are positive and awesome. Anion, an means without or against, so negatively charged. So positively, they're filtered more easily, and that is because of the Donen effect, which we are going to explore again right now. I'm starting to speak louder to hopefully let you know that this is really important. You're expected to know this not just for your exam and your boards, but really for all of your other classes. This is really important. And the Donen effect is part of these forces that keep fluids in their place where they're supposed to be. So that we have a plasma that is mainly positively charged. We've got proteins that are negatively charged. Again, that's called the Donen effect. And as long as there's proteins, remember, proteins are negatively charged. And if the rest of the plasma components are mainly positively charged, negative and positive, they're attracted. So just having proteins hanging around in the bloodstream is enough of an attractive force to keep all the plasma components within blood vessels. And these forces are very important in also driving filtration. So what we're looking at here, again, we've got the afferent arteriole, so the arteriole leading to the glomerulus. And we have the efferent arteriole leading away from the glomerulus. So blood is coming in, forced into the glomerulus at a high pressure, and then it has to leave. These arterioles, they have these sphincters on them. You can see them in these pictures, which can help modify the tension in these vessels to make sure that we have a stable net pressure that isn't changed too dramatically by things like exercise or sympathetic outflow. Remember that whenever blood pressure goes up in the body, we wouldn't want our filtration rate to go up so high in the kidneys at the same rate that systemic blood pressure would go up. In that case, it would mean that you would just be peeing continually as you went for a run because your force or rate of filtration would be so high because of a high pressure in the glomerulus. So these arterioles, they respond to products that help modify the rate of filtration depending on what's going on in the rest of the body. Okay, so if we go through this Starling Force picture here, again, these are things that you need to know. This is something you shouldn't forget. You don't necessarily have to know the numbers, but you should definitely understand the premise of things like the Donen effect, which means proteins are negatively charged, plasma is positively charged, negative and positive are attracted, which means fluid stays in leaky places because of this magnetic attraction. So there's a couple of different pressures we have to take into account when we think about whether or not fluids or anything is really going to take the chance to leave the glomerulus and enter Bowman's capsule. The predominant pressure here, this outward pressure, is called the glomerular hydrostatic pressure. Hydro is water, and static is location. So if something is static, it usually means it's not moving, but it's just a word for location. So glomerular hydrostatic pressure means the pressure that's exerted by fluid in the glomerulus. So this is a pressure of about 60 millimeters mercury. So fluid enters the glomerulus and its pressure, just the pressure of fluid, is really high. So in the same way, you would turn on a garden hose, draw a little hose here. If you put your thumb, there's your thumb, over half of it, that's a nail, that looks nice. That means the pressure is going to go way up, right? The water is going to squirt out. That's hydrostatic pressure. So that's the force of the fluid, in this case within the hose, that's going to favor outward pressure. If you turned your hose on really, really low and put your thumb over it, and it's just a couple of drops, it's not going to be the same force at which water exits. It might just like drip out a little bit. So it's a high pressure which favors it leaving. In the same way, water will leave even when you try to cover up the hose with your thumb. Then there's some opposing pressures. The first one is the opposing pressure of the fluid in Bowman's capsule. So there's some fluid here. There's not as much because it's just a little bit of fluid that's being squeezed out of the glomerulus. I wrote over that. So we have the pressure of fluid in Bowman's capsule, which is relatively low because it's just the leftover thing squeezed out of the bloodstream. So if we look at just these levels, in the glomerulus, the pressure of fluid is 60 millimeters mercury. In Bowman's capsule, the pressure of fluid is 18 millimeters mercury. 60 is a much higher pressure than 18, which means that this is not enough pressure to oppose fluid leaving. So this is the predominant pressure right here. Then we have, I'm going to get rid of my writing in just a second. So the final pressure is going to be oncotic colloid pressure. And this is where we get back into the Donnan effect. Oncotic colloid means the pressure exerted by the colloids in the blood, which are proteins. So remember, proteins are negatively charged where plasma constituents are positively charged. So those two things are magnetically attracted to one another. They cling to one another. Stage 5 clinger right here at the level of the glomerulus. So the proteins don't want the water to leave because they're attracted to them. But this is a really high pressure, there's holes here, and because the pressure of the fluid, still 60 millimeters mercury, is higher than the pressure exerted by the proteins clinging to the fluid, asking it not to leave, it's still going to be overcome because 60 is greater than 32. If you think about what's going on in Bowman's capsule, there's no proteins in here, right? Because proteins are too big, hopefully, to make it through those holes. So we don't have a colloid pressure out here in Bowman's capsule. We just have a hydrostatic pressure. Okay, so Bowman's space, this little space between the glomerular capillaries and Bowman's capsule, again, it would be like filling up a water balloon with some water. You're getting ready to smack somebody with it, but before you do, you just place your fist, that looks nice, into the water balloon. So you hold the water balloon in one hand. Here's one hand. I actually draw very well in other situations. And then with your other hand, you just put your fist into the water balloon. And then it's going to make a little cave for your hand. Your hand won't be inside the water balloon, but it's pretty much as close as it can get. That's the relationship between the glomerulus and Bowman's capsule. So there's just a little bit of space in between them. And there should be no proteins there, so we're not going to have any proteins attracting fluid from the other side. So the predominant pressure is just the water, just the fluid inside of the glomerulus, which is very high, and that is going to favor things leaving. If that wasn't the predominant pressure, we would never urinate. So it's the predominant pressure, and it favors fluid filtration. That is called the glomerular filtration rate. When you have these nice stable pressures, there is a normal rate at which things move through here, which again, you can alter depending upon what's going on in the body. So this is just to tie that up. When there is sympathetic stimulation, so you're exercising or blood pressure goes up for any other reason, there's going to be glomerular filtration rate differences usually. Because we have, so here's renal blood flow, so the blood flowing through the kidneys, and then the rate at which that fluid leaves the glomerulus. So when you do something like exercise, right, so sympathetic nerves, they're vasoconstricting, they release norepinephrine. If the extracellular fluid volume decreases and we go into sympathetic outflow, so that could even be because you're sweating and you're losing some of the fluid. That decreases the rate at which we lose fluids through the glomerulus, which makes sense. If your extracellular fluid volume goes down and you have less overall fluid in your plasma, you don't want to be peeing out more fluid. You don't want more blood to go into the kidneys and increase the pressure for filtration. Same thing you'll learn about in blood pressure. Angiotensin II is released anytime the blood pressure needs to go up. It is designed to save you. And it's released when there's a decrease in extracellular fluid volume as well. So if we're trying to save the body, like let's say this person went into shock and now the organs aren't getting the blood flow that they need, we also need to decrease the rate at which we filter things through the kidneys and the pressure of blood within the kidneys. Endothelin is a product that is released when there's increased stretch in muscles, like in vessels, when angiotensin II is around. Bradykinin, we know, is associated with the pain response. Epinephrine, lower extracellular volume, that will decrease the glomerular filtration rate as well. Vasodilators, though, so whatever is going on systemically, if the blood pressure is going up, these are all vasoconstrictors. If the systemic blood pressure goes up, the pressure in the kidneys is going to have to go down. Because otherwise, if this doesn't go down, this will be matched, meaning renal blood flow will be matched with systemic blood flow, which means you're going to lose all this extra water, which is not going to be consistent with life. And we try not to do things that are going to kill us at the level of the body. So in that same way, vasodilators lower blood pressure. They make vessels dilate. So some things are going to then have to increase the rate of glomerular filtration because we still need to make urine, make a concentrated urine, even when the blood pressure is low. Prostaglandins are formed in inflammation. They can be released when there's decreased extracellular fluid volume, a lot of shear stress, which is just the stress of blood components scratching against the blood vessel wall. Also, angiotensin II can make vasodilator products called prostaglandins pop up. There's no real change in prostaglandin activity on the glomerular filtration rate, but there is an increase in renal blood flow with prostaglandins. Because prostaglandins are associated with injury and disease usually. That's what aspirin and NSAIDs block, for example. And you've probably heard that if you take too many of those, it damages your kidneys. Maybe you've heard that before. That's because the blood flow to the kidneys increase so that you can excrete these products of things like damaged cells. Nitric oxide is a local vasodilator. It's released in shear stress to help keep the vessels from being damaged. When acetylcholine's released, histamine, bradykinin, just ATP. Nitric oxide's a really potent vasodilator. It really lowers the blood pressure. So when we have potent lowering of the blood pressure, we will have the opposite effect. We'll have an increase in the GFR and renal blood flow so that we keep producing urine. Same thing with bradykinin, that's our pain response and chemical pain that is released anytime there is a breakdown product of damaged cells like prostaglandins, angiotensin converting enzyme, ACE, you've probably heard ACE inhibitors. ACE inhibitors block the conversion of Ang1 into angiotensin 2, which people take therapeutically when they have really high blood pressure. Because Ang2 is only supposed to be around when you're dying. It's supposed to keep you alive. So an ACE inhibitor would mean that you're lowering angiotensin 2 and then lowering the blood pressure. So anyways, bradykinin can be stimulated by all those things. It's a vasodilator. So those products will increase renal blood flow and glomerular filtration rate. Then there's two fun little hormones, atrial natriuretic peptide, which is released by cells in the heart in response to levels of sodium. We also have brain-derived natriuretic peptide, also released when there's high levels of sodium. And those things are released when the extracellular fluid volume goes up. So that, remember, where sodium goes, water follows. So you have a really salty meal, you've got tons of salt in your body now. These products will be released to help counteract this effect by making you pee out the sodium and the water, hopefully. So that means we're going to have an increase in glomerular filtration rate and an increase in renal blood flow. So the bottom line is that anytime there's systemic raise in the blood pressure, we're generally going to decrease kidney filtration and activity. Anytime there's a decrease systemically in blood pressure, we're going to have to generally increase the level of filtration in the kidneys. So those are things that modify that all of the time. The other thing that the kidney does is do that itself. It's called autoregulation, and it's a self-feedback mechanism. And this is what affects the arterioles, the afferent and efferent arterioles, which have their own sphincters associated with them. When there are changes to the systemic blood flow, we already talked about the external products that can cause changes to the GFR. There's also this internal mechanism. So the JG cells, the juxtaglomerular cells, they sense blood pressure. They specifically sense things like ischemia. So when blood pressure is low, juxtaglomerular cells, they secrete a product called renin. And renin is part of the renin-angiotensin-aldosterone system. So renin is cleaved by a product called angiotensinogen into Ang1, which is then cleaved by angiotensin converting enzyme, ACE, into Ang2. And that's going to raise the systemic blood pressure. So that's the juxtaglomerular cell's job. It's to sense levels of blood flow and sodium levels as well, to signal to the body that blood flow to the kidneys is down and we're going to need to raise the blood pressure. And when the blood pressure is raised, that will affect filtration. Also, the macula densa cells we talked about really briefly. You can look earlier in the slides too. Those are responsible for monitoring things like sodium levels, which will trigger the release of other products to affect the activity of things like the efferent arteriole and afferent arteriole. So going through this system, it's called the Tubuloglomerular Feedback loop. So these are our two cells we just mentioned. We've got macula densa, and then we've got juxtaglomerular cells. So here is this autoregulation center. So first we have a decrease in systemic blood pressure, a decrease in arterial pressure. Okay. When that happens, the glomerular hydrostatic pressure is going to go down, so there's going to be less pressure in those blood vessels, which means the rate and pressure of things leaving the glomerulus is going to go down. What that means is that you're going to have more buildup of byproducts in the blood because they're not being filtered. So when the GFR is low, so we have more retention of filtrate that we would normally put in the urine, that is going to trigger the macula densa cells, which look at levels of sodium chloride. The macula densa cells are going to trigger the other release of renin, angiotensin, and then decrease resistance. We can also have a direct decrease in what the afferent arteriole is doing. So we have efferent arteriole, afferent arteriole. So remember, macula densa, looking at sodium chloride, renin, angiotensin 2. Efferent arteriole resistance. So if I drew this out, so here's afferent, here's a glomerulus, here's efferent arteriole. If I increase efferent resistance, that means we're going to contract the sphincter in the efferent arteriole, which is going to raise the GFR. Does that make sense? So if the GFR is down, we can constrict this efferent sphincter, raise the GFR. We can also lower the resistance of the afferent arteriole, meaning we can dilate the afferent arteriole, letting more fluid in. At the same time, we're constricting the efferent arteriole. That's going to increase the pressure and increase the glomerular filtration rate. So these are the autoregulatory bodies, just the sphincters, the afferent and efferent arteriole, and they are going to be dependent on these macula densa cells. The JG cells, it's just their job to release renin when they're either triggered by the macula densa cells or they're triggered by a really strong decrease in blood pressure. They sense ischemia in the kidneys, so they're triggered when there's a lack of blood flow. So in high sodium cases, the GFR will be really quick because there's a high level of blood pressure. Where sodium goes, water follows. A local vasodilator in all systems in the body is called adenosine. It decreases the resistance of all vessels, and it also helps constrict the afferent arteriole, which is our inlet pathway. So there's less blood in. If there's less blood in, there's going to be less filtration. So we don't have constant urination when the blood pressure is really high. So the GFR will go down. If there's low sodium states, so that means the blood pressure drops. So just like we talked about in this picture, the macula densa cells, they sense the lowered levels of sodium chloride, and then they help trigger the JG cells to release renin, which will be converted into angiotensin II. Constriction of the efferent arteriole, so that's blocking the outlet door. That's going to raise the pressure of blood in the glomerulus and then ultimately increase the rate of filtration. So we'll have an increase in the GFR. There's tons of review questions about this if you look up even the med school boards, because this is always part of the med school boards, because it's a perfect way to test if somebody understands physiology or not. So there'll be questions like, if we want to increase the GFR, what would be the best action by the body? Would we increase the constriction of the afferent arteriole or decrease it, or vice versa? So these things will be modifying the tension in those sphincters, which will help maintain a steady GFR and steady rate of urination. Woo! I hope you liked it. I love the kidneys. Back for more.